Hello, my name is Cindy Lopez, and today I will be presenting and interpreting data using reverse transcriptase polymerase chain reaction, RT-PCR. So what is RT-PCR? RT-PCR uses an enzyme called reverse transcriptase, which is used to change a specific RNA sequence into a matching DNA sequence. That new DNA is then amplified by DNA polymerase using PCR, and then that newly amplified DNA can help then determine or find if a specific mRNA molecule is being produced by a gene. It's generally used to look for any type of changes in the genes or chromosome, and is also used to look for any activation of certain genes. So there are two types of RT-PCRs that I came across during my investigation of what RT-PCR is. The first one is called one-step RT-PCR. It is generally used in one vessel, and it basically puts the reverse transcriptase and the PCRs done at the same time. It does use this gene-specific primer, which if you're looking for a specific gene, that's a plus for you right there. And it's typically used for only a small number of assays. It is also done very quickly. The cons to this type of PCR is that it does impact yield of the copied DNA that comes out of this reaction, and generally you need more samples of your RNA if you ever need to repeat this reaction. The second is called two-step RT-PCR. Pros to this is that it can be carried out in two separate vessels, and it uses a variety of different kinds of primers. So basically how it works is that you get the first step, you put in all of your elements in, including the reverse transcriptase, you run it, and then you put the primers in, the specific primers if you need to, and then you run the PCR, and then you get a high yield of the different DNA species that are in there. This is also good for any type of long-term storage compared to the one-step RT-PCR, because you just get so much more out of it. The cons to this is that it is more time-consuming, so it takes a lot longer compared to the one-step. And there is a high risk of variability and cross-contamination, since you're doing it all in a two-step, and if you're doing multiple at the same time, you have that high risk of cross-contamination. So I found an article called Reverse Transcriptase Polymerase Chain Reaction versus Chest-Computed Tomography for Detecting Early Symptoms of COVID-19. Basically, this article was trying to compare to see if the RT-PCR was just as reliable as a CT scan on the early detection of COVID-19. This was published during COVID, so it was a pretty important paper at the time, I'm sure. The experimental design, they took data from, I believe, 5 different databases, and they were all included in a systematic review. Approximately 1,200 patients with COVID-19 were evaluated, so these were all positive cases. 1,045 had tomographic findings, so like within the CT scans, something came up that led to the realization that this patient had COVID-19. And then 755 of these patients also came back positive with RT-PCR positive for COVID-19. So in this table here I've provided, comes straight from the study. It basically took, like I said before, those 5 studies, individual studies that were performed. So you have I et al., Burnham et al., Fang et al., He et al., and Long et al. They list all the number of patients that they saw. They categorized who had early detection of COVID-19 in RT-PCR and who had early detection of positive COVID-19 using a CT scan. And then in this last column here, where it says comments, is where they're comparing if patients who had a negative early RT-PCR test, as in the results came back negative, later or had a positive CT scan when done around the same time. And it kind of shows here, well, if you read down this list here, it basically summarizes saying that most of the patients that were positive with COVID-19 came back with a negative result with early detection if they only used the RT-PCR. But if those same patients, for the most part, went straight to go get a CT scan, they had a higher chance of being tested as positive. So basically what they're saying here, and basically I put in a D here where I interpret the data, overall CT is more reliable and sensitive at the sensitivity and detection of COVID-19. So in conclusion of this paper, they're saying that for early detection of COVID-19, RT-PCR is not as reliable as compared to a CT scan. So for our presentation, we needed to interpret some data, and that's just what I'm about to do here in this slide. So FDN is a molecular compound, and as you look here on the x-axis, the concentration of FDN increases steadily. Here, BAX, BAD, and BCL2 are going to be genes, and you can see here that as the concentration of FDN increases, the expression of BAX and BAD increases. It is significantly increased at the 200 micromoles for both BAX and BAD, but for BCL2, as you increase the concentration of FDN, BCL2 significantly decreases, so its concentration goes down. And that is actually my interpretation of this data. Finally, this is my reference slide. If you guys have any questions, please let me know and I'll be able to answer them for you. Thank you for watching.